So what I want to talk about today is one of our products, APO10, which is a hexameric linking 6 Fas ligands together in a what we call mega Fas ligand. And we see this as a new immune checkpoint inhibitor. And we are entering phase 2 with our product in multiple myeloma. The Fas system is one of the 2 important systems that the tumor cells are being killed via the T cells. We have the Fas system and we have the perforin or the granulocyte mediated cell kill. And what we've done is basically make a hexameric of the Fas ligand. When you have 6 Fas ligands tied together, the tumor cell seizes as a cell membrane bound Fas, and it's a very strong apoptotic signal. So we have we worked that into a product. It's a big molecule. It has the size of 180 kilodalton, so it's almost like 1.5 antibody. And here you see basically how we have worked on different programs, but ending up with that a hexameric form of the Fas ligand is very cytotoxic and fully capable of you can say activating apoptosis. A lot of Fas is also available in a soluble form, and if you have Fas in 3 trimers, they can even be antagonizing the cytotoxicity of the T cell. So what we see is that we produced you can say an artificial T cell, a system that can bring cell killing into the tumors by the T cell mechanism. So basically, you can say that with a 3 trimer, we don't see any activation of apoptosis. If we do the hexameric form, we see very strong activation. So this is kind of how APO10 looks. And the tumor cell reads this as a membrane-bound Fas, leading to a strong apoptotic signal. In a way, you can see that the system, the apoptotic program here is, you can actually see APO10 as a highly targeted cytotoxic. And as such, it could resemble antibodies that are binding to an antigen and carrying a drug conjugate, but this is all in one. We are, you can say, if the Fas receptor is there and the downstream elements are available, the cell will be killed by this compound. We call it Fast and Furious, which is of course inspired by some movies. Basically, we have seen it's effective in a number of cell lines, and we do see Fas receptors in several tumors, in breast cancer, in ovarian cancer, in multiple myeloma. Fas receptors are very frequently there. And we're also getting a profile of Fas cell kill when we look at the NCI60, which is a program that we use to identify responding patients. I'll talk to that later. We've seen not only in vitro data, but also in vivo data, we can see that it works. There's always been the problem about Fas. Is Fas actually too toxic to develop into a cancer drug? We do have the DES receptors on some of our normal cells, for instance, the liver cells, but we do see here a clear window of opportunity where we are able to treat this mouse with a non-small cell lung cancer. This is xenografts, but we also have syngeneic mice where we are able to kill the tumor. So it's more toxic to the tumor cells than to the mouse model. We have here glioma. So that in vivo, we have a study with glioma where again we are increasing the survival of the mice with our APO10. In ovarian carcinoma, we also have in vivo good data. And in multiple myeloma, we have demonstrated here with a syngeneic model that we actually have more potency than with Velcade, the bortezomib product that is used for multiple myeloma. We use our drug response predictor to develop our product into the most likely responding patients so that we can get into the indications where we have the highest likelihood of response. And when we look at our database and look at where do we see the highest likelihood of sensitive patients, we have certain diseases where we go for, and where we have, you can say, the highest frequency of sensitive multigenetic. We basically use a multigenetic, a messenger RNA profile of more than 200 genes, where we're looking at the potential for moving the product forward. This is what we do in Oncology Venture generally, is we take products and move them forward by this technology. So we in-license products that has shown activity in patients and use our genomic expression profile to increase the response rate and have products approved. Apo10 is, you can say, in many hematological diseases, there is it could be a very interesting product, but also in some of the solid tumors. Picking the right patients, the drug response prediction, I don't want to go into that in this talk today, but this is our core technology, utilizing patterns in drug sensitivity, and that could be, for instance, NCI, where reflex mechanism of action of the product, and we correlate, you can say, the messenger RNA in the in vitro systems. And in order to make this clinically relevant, we have to, you can say, filter through a clinical relevant path or filter, and we have 3,200 patients where we ensure that you can say, the pattern in expression is making sense, so we can get rid of the or the background noise and only look at pathways that are clinically relevant. We also think that Apo10, in addition to being directly cytotoxic, also has this, you can say, translational link between the innate and the adaptive immune system. And we are here in the, you can say, in the sort of moving to the adaptive system where the fast system is available. And what we expect that Apo10 can help us with is to move into, you can say, non-immunogenic solid tumors, and in essence, go in and turn cold tumors hot, start a approach which looks like a T-cell approach, and then having, you basically say that expecting a cascade of next level events, and basically you could say getting a direct apoptosis via fast signaling, and then also stimulate a second wave of immune attack. So we do see this as a, you can say, a checkpoint, blocking inhibitor, basically being able to attack the tumors as if it was T cells. We don't have tumor infiltrating T cells in many breast cancer tumors, in many tumors, and we don't know how to get the T cells in. And with this, we have actually the most important T cell killing mechanism that we can add directly to our tumors. So we see that there is a lot of things that could be problematic about how we get these tumor cells into a immunogenic, and all these are kind of irrelevant to how we see Apo10 moving forward. We are in multiple myeloma, and we see it's an interesting place to be, where it is difficult to see how you can say the PD-1, PD-L1 blockade can actually move patients into sensitivity. And we have just started now a phase 1/2 trial in multiple myeloma, where we have used our drug response predictors, so that we are only including the high likely responders, but we also, of course, aim to look at the you can say turning tumors hot in this setting. But right now we are doing the Apo10 as you can say, as an anti-cancer agent directly into multiple myeloma patients that are selected for high sensitivity to our product. We have screened 50 patients in 4 different hematological centers. They're very excited about this product, and we look forward to move it forward. It has already been in a phase 1 study where it was in all comers. We never used the drug response prediction method, so we of course have now optimism in how this is going to move forward. And next step is also to utilize some of the fast synergy that we know is there. There is a lot of drugs out there, that actually increase fast sensitivity. So we've seen it with bortezomib, which is you can say typical, or would be a good product in multiple myeloma. Of course, this is Velcade, and other of these proteasome inhibitors could go in, but also interferon, HDACs, many cytotoxics, irinotecan, cisplatin, gemcitabine. I saw the other day increase the cancer cells to fast ligand cytotoxicity. So we believe that we're in an interesting situation with a product that by itself is highly targeted, going for the fast receptor, but also is able to elicit immune, you can say, turning hot tumor, cold tumors into immunogenic tumors.